I like this image because it gives you a pretty good idea of the shapes of the cells that we're going to be talking about. As we talked about in the last section, there are categories of epithelial cell types, and the way that we will categorize and name them will be based on the number of layers of cells as well as the shape of the cells. When you talk about simple squamous epithelium, and please always write ET when you are writing about cell types, epithelial cell types specifically, because if you do not and do not understand, or you know that you are talking about epithelial tissue. So simple squamous ET. If you take a look at these guys, they look like fried eggs. So it looks like you have a whole bunch of fried eggs all connected together. Simple cuboidal epithelium is going to be a single layer of cube-shaped cells. When you talk about simple columnar epithelial, you are talking about these taller shoebox-shaped cells. When you look at these images, you can see that there is no space and no stuff in between the cells. So when we talk about tightly packed cells, there you have it. Simple, when you're talking about epithelial cells, means a single layer. Stratified means multiple layers. So simple indicates a single layer and stratified indicates multiple layers. The other reason I like this image is because it reminds you that epithelial tissues have a basal membrane which will connect those tissues to the underlying connective tissue. The first type of epithelium we're going to talk about is simple squamous epithelium. And again, those are those sort of fried egg shaped cells. Very flat, very thin. So you're going to find these in areas where you need to get fusion happening quickly. So where would you find them? These will be found in the alveoli, and the picture on the lower right is going to be an alveolus, very thin, and gas exchange will happen easily. The image on the lower left, this guy right here, is a glomerulus that you're going to find in the kidneys. And if you take a look at this thin layer here, that thin layer is going to be simple squamous epithelium. And sure enough, material can move through that pretty quickly and easily by diffusion. Again, these are the thinnest tissue of the body. And why do we care? Because it's going to allow for pretty rapid transport via diffusion. And this is classically simple squamous epithelium. And this will be found in the lungs, and it is going to be part of the alveoli of the lungs. Hugely important to have quick, rapid diffusion of gases. And another shot of the alveoli. So here's a question. These are supposed to be a single layer of stratified squamous epithelium. How come we have this? Or this? I guarantee you this is simple squamous epithelium. Think about that. And remind me to answer that in class. And what about this? Simple squamous epithelium. That is looking superior to inferior. You're actually looking down at the tissue, you're not looking at a transverse section. Now, simple cuboidal epithelium. So you have a single layer of these cube-shaped cells. These guys are going to form ducts as well as kidney tubules. They're also going to be secretory cells that you're going to find in exocrine glands and in kidney tubules. These are going to form the walls of kidney tubules. So these guys have more cytoplasm, but not as much as simple columnar epithelium, which means that you can still move material through them fairly quickly, but you don't have a lot of cytoplasm which you would require for processing. So these show you simple cuboidal epithelium. This is going to be the wall of a kidney tubule, and here's another one. And what will you have in between? Sure enough, you will have the basement membrane. You're right. And here we go with another layer of that simple cuboidal epithelium. Simple columnar epithelium, you will have those toral shoebox-shaped cells. Notice that you have a lot more cytoplasm, and that will be important for things like processing nutrients. You may also have a couple of modifications to the apical surface, which would be cilia or microvilli, depending on where these cells are found. This shows you a lovely diagram or micrograph of simple cuboidal epithelium. Look at how tall those cells are. You see how the nuclei are lined up all along towards the bottom of the cell? That is classically simple columnar epithelium. You can see that the nuclei are all sort of located along the bottom of that also. This shows you some microvilli. You're going to find these in the digestive tract. And this is a micrograph of microvilli. Goblet cells are going to be very important to know about. In various parts of the body, you are going to need mucus, and especially in places like the digestive system. You're going to need mucus because as you move material along the length of the digestive tract, there's a lot of wear and tear that goes on. So the mucus actually provides lubrication, which means that the material can move more efficiently and more rapidly and do less damage to the walls of the GI tract. This shows you simple columnar epithelium with cilia. Now, we do have a couple of sort of these outlier epithelial tissues. One of them is going to be pseudostratified columnar epithelium, and in this case it's ciliated, so you get the word ciliated in there. Pseudostratified columnar epithelium is the major lining of the respiratory tract. And it will have goblet cells associated with it, and it will also have cilia. And the reason that we want goblet cells for mucus and cilia for moving material across the surface of the membrane is because you need to trap any sort of dust particles or particulate matter that has entered the respiratory tract and move that out of the respiratory tract before the dust or particles get down into the lungs. I want you to think about this term, the mucociliary escalator. Mucociliary escalator. And let's talk about what that is in class when we're meeting in person. When you look at pseudostratified columnar epithelium, it's called pseudostratified even though it's a single layer. It's called pseudostratified because it's sort of this weird, funky looking tissue. The nuclei are going to be scattered all over the place. You'll remember in the simple columnar epithelium, the nuclei were all sort of lined up towards the bottom of the cell. In the pseudostratified columnar epithelium, it looks like multiple layers, but it really is only one. And in fact, all of those layers, all of those cells do attach to the basement membrane. It just looks kind of weird. And in that picture on the right, you can definitely see the cilia along the apical surface of the tissue. Whoops. There's cilia. Up close and personal. Now, stratified squamous epithelium is going to be important. You have those sort of flattened egg-shaped cells, except you have multiple layers of them. When you talk about stratified squamous epithelium, you are usually or often talking about the epidermis of the skin. Now, the skin is part of the integument, and you're also going to find stratified squamous epithelium in the mucous linings of the mouth, the oral cavity, as well as the esophagus, in the anus, and in the vagina. When you talk about the stratified squamous epithelium that you find externally, like part of the skin, you're going to see that it can be keratinized or keratinized. I don't care how you pronounce it, as long as you can spell it in a fashion that I can actually recognize it. The skin, the epidermis, will make this waterproofing protein, which is known as keratin. And you'll also see this. Look at where the basement membrane is. It's going to be the deeper aspect of that tissue. When you look at the cells that are just superficial to the basement membrane, they actually kind of almost look like simple cuboidal cells, but they are not. They look rounder and plumper because they have just undergone mitosis, and as mitosis continues to occur, cells get pushed up higher and higher and higher and higher through those layers until they are basically just these flat, anucleated packages of keratin. As they move away from the basement membrane, they are moving away from the blood supply. Let me repeat that. As they move away from the basement membrane, because mitosis is occurring, they are moving away from the blood supply. So you are more likely to have cells that are going to die and slough off. Sure enough, that's what happens. The old skin cells are going to exfoliate from the surface. This shows you stratified squamous epithelium. And this happens to have keratin in it. And this also shows you stratified squamous epithelium. You can see the basement membrane is going to be in through here. These cells look plump and juicy and round because they have just undergone mitosis. And as they are pushed further and further and further away up towards the surface, they become flatter and flatter and flatter until, like here, they're actually sloughing off. These are all dead cells that are going to be sloughed off. Last but certainly not least, we have transitional epithelium. And transitional epithelium is another one of those outliers which you're going to find in the urinary bladder. So when the bladder is empty, these cells look more like stratified cuboidal type cells, but they're not. They are transitional epithelium. As the bladder fills, these cells have the ability to flatten and expand because they are being stretched as the bladder fills. I would not put any images of these guys on a lab practical because it is just too hard to tell what they are. So here they are in the wall of the urinary bladder. It shows you what they look like when the bladder is relaxed, and that shows you what they look like when the bladder is full. Oops. And this shows you the same thing. The upper picture is the relaxed and empty bladder, and the lower one is the full and therefore stretched bladder. We need to talk briefly about glandular epithelium. The glands are going to be lined by epithelial tissue, and there are going to be several different types. When you talk about merocrine glands, for example, the salivary glands, they are producing and releasing a secretion, in this case saliva. When you talk about apocrine glands, not only are the glands producing a secretion, actually part of the gland is being pinched off and released with that secretion. And then when you talk about holocrine glands, and an example of that is sebaceous glands, which we'll talk more about when we get to the integumentary system, the actual cell dies and becomes the secretory product. Really just fascinating. This gives you an overview and a review of the different types of epithelial tissue and more as it happens.